Good afternoon, everybody. Welcome back to Virology. Today we're going to talk about some infectious agents that don't go anywhere else in this course. They don't fit in, but they're very interesting and you can learn a lot from them. So I call this unusual infectious agents. And this lecture will address a fundamental question in virology. What is the minimum genome size needed to sustain an infectious agent? Or could an infectious agent even exist without a genome? Well, we're going to talk about viroids, satellites, and prions. These provide answers to these questions. We'll start with viroids. These are circular, single-stranded RNAs from 120 to 475 nucleotides in length. And they're extensively base paired, as you can see here. And if you look at them in an electron microscope, they appear as a rod because they're extensively double-stranded. They have no protective coat. There's no capsid. It's just naked RNA. Yet they migrate from host to host. And they replicate when introduced into plants, actually. So the hosts for these were originally found to be plants. Now it's expanded quite a bit. But they migrate from plant to plant. No capsid, just naked RNA, and they replicate. And there are two families, Viroidae. So remember, the viruses are Viridae, but the viroids are Viroidae, different name. These replicate in the nucleus, and the avsun Viroidae replicate in chloroplasts. Some of these viroids are ribozymes. The ribozyme is an enzyme that consists only of RNA. And for those that have ribozymes, this activity is essential for replication. Now, there's a simple way to recognize or realize what viroids are. Compare them to viruses. Viruses are parasites of the host translation machinery, right? They need to make mRNA that can be translated into proteins by the host. Viroids don't encode protein. So they are parasites of the host transcription machinery. They need to be copied by host enzymes, not translation because they don't encode protein. So the first viroid discovered by T.O. Diener here, who died in 2023 at 102. Good riddance. He discovered potato spindle tuber viroid. And this was, as I discovered in 1967, the first viroid to be discovered. So it became the prototype for these viroids. He's the one who named them viroids. It is 359 nucleotides in length. And PTSVD, so these are the way these are named. VD means viroid, and then the rest comes from the name. Some of them have effects on crops, as you can see here. These plants are healthy on the left, and then different strains of potato spindle tuber viroid can cause different amounts of growth stunting. So these are agriculturally very important, obviously, because you can't grow proper crops when the plants are small. It's very important. So many farmers use seeds from the same companies. There are a few big companies that sell you seeds. And so you have to make sure your seeds are free of viroids, because if you plant seeds with viroids in your soil, you'll grow up plants with viroids and they'll have stunting like this. So companies are very careful to screen seeds for viroids and make viroid-free seed stocks. So I have some favorite viroids because the names are very interesting. So the first one is the Gadang coconut viroid. This causes a lethal disease of coconut palms. That's a coconut palm there. And you can see it's brown because it has a viroid infection. So if you like pina coladas, you'll be sad at this because you need coconuts for pina coladas. Then there's the hop latent viroid, which causes no symptoms in the hop plant. Here's the hop plant. Of course, hops are used for making beer. So if you like beer, you're happy that this doesn't cause any disease. And then there's the apple scar skin viroid. I mean, they just name it according to the disease. It's great. I did a podcast this morning with a plant virologist in Europe, and he told us the name of some of his viruses, and he giggled because they're so cool like these. He giggled at the names of his viruses. Apple scar skin viroid gives apples mild symptoms. They look bad, but they taste good. So that's an apple there with this viroid. You know, it's blotchy. You're not likely to see these in supermarkets. They're probably not going to sell them. And so if they did, people wouldn't buy them probably. Would you buy an apple that looked like that? You say, what's wrong with this apple? I never saw something like this before. But they're perfectly good, and viroids cannot hurt you. Lots of different viroids. So viroids have functional regions. So these are the two kinds of viroids. The pos B viroidae on the top. That's potato spindle is a member of that. And then the avsun viroidae. Peach latent mosaic viroid is an example. So the pos B viroidae are rods with a central conserved region. There's a region on the left that's important for pathogenesis. And then the avsun viroidae are more extensively branched. And you can see these have a ribozyme. So these shaded bases will self-cleave themselves. That's the feature of a ribozyme. It can cleave itself. It's self-cleaving RNA, which it was present in the RNA world most likely. So these are relics of the RNA world. Now, viroids can spread both vertically and horizontally in plants via pollen. So a viroid gets into a pollen. Here's vertical transmission on the left. Here's the infected pollen. It comes from another plant. It's got a viroid in it. The viroid can travel down the pollen tube into the ovule, and then the new seedling will be infected. So that's vertical transmission. And then, of course, horizontal is from other plants. The same thing happens. The pollen is infected. It goes to this plant and invades the plant. Not transmitted vertically in that scenario, but it's transmitted horizontally. So how do they replicate? They're copied by host RNA polymerase II. So they're the one exception to host enzymes being able to copy RNA genomes. But host pol II, of course, normally recognizes DNA templates. It's the enzyme that makes mRNA in our cells. So one of the groups of viroids, as I said, the avsun viroidae, they have this ribozyme, and there's one kind of ribozyme called a hammerhead. Looks like a hammerhead. And this ribozyme activity, by the way, the ability to cleave itself in the absence of protein was discovered in 1981 and got the Nobel Prize for that discovery. And what happens is these viroids, as you'll see, make concatemers, which are then cleaved by the ribozymes. And other viroids are cleaved by host nuclear enzymes. So let's look at the pos B viroidae, how they replicate. This is a plant cell. It has a cell wall that has plasmodesmata that connect all the cells. So all the cells are connected. Things can move from cell to cell very readily in plants. And it has a nucleus, of course. So the viroid is shown coming in here, the circular single-stranded RNA. It goes in the nucleus, and it's copied by pol II into an antigenome, back to the genome. And you can see there are concatemers made. We're showing two for clarity. These are then cleaved in the nucleolus into monomers, which circularize and get exported, and then they can move to other cells in the plants via these plasmodesmata. So the viroid spreads throughout the plant and affects all the cells. The avsun viroidae replicate in the chloroplast. So we don't have a nucleus shown. This is the chloroplast. It's bigger than it should be, but it's there. And these avsun viroidae go in the chloroplast, where they are copied by chloroplast enzymes, and again, into concatenamers, which are cleaved by the hammerhead ribozyme before they move to the adjacent cell. So the difference is in what is copying these ribozymes, these viroids, and what is cleaving them. Where did they come from? Well, when we discovered them in the 60s, we used to use wild crops to produce modern crops. We bred wild crops until they had the properties that we desired, and then we kept interbreeding the modern crops. But at some point, those wild crops had viroid contaminants in them. And so someone found, T.O. Diener saw that the plants weren't growing properly, and so he discovered the viroid as the cause. And these, as I said, these are probably relics of an RNA world where RNA was self-replicating and could cut itself, the precursor to maybe cells and viruses. And the problem with viroids is that everybody in the world uses identical plants. Basically, they use the same seeds, and so if you get a contaminated seed, you will all have viroids. And in addition to moving around by pollen, viroids can be transmitted mechanically. Contaminated farm equipment or machinery, farmers' hands, touching plant to plant can transfer it, the viroids. Very hard to control. So now, how do they cause disease, right? Some of them cause overt disease, and that's why we worry about them. Basically, when the viroid comes into the plant cell, it's chopped up by the microRNA system, which we've talked about before. And you get these small 21 to 24 nucleotide RNAs called siRNAs, small interfering RNAs. And those seem to silence host genes, plant genes. And the silencing of the plant genes causes the disease. So the siRNA production is a defense by the plant to try and chop up this viroid, but it turns out that some of those small RNAs can shut down host genes and cause disease. And these siRNAs map to this pathogenicity modulating domain of the viroid. Right? So the siRNAs are made from the viroid with the idea that they will hybridize to new viroids that are incoming and destroy them, but in fact some of them turn off host genes and cause disease. Now, more recently, people have found viroid-like circular RNAs in metatranscriptomes from all over the world. A metatranscriptome is a collection of sequence data. So people take an environmental sample like water or soil and extract RNA and sequence all the transcripts. And you can, these are put in public databases, and you can search them, and people have found lots of viroid-like sequences. And they're all over the world, as you can see here, and they are in different environments. So most of them seem to be in the soil. So remember, these were originally just thought to be plant pathogens, but now they're in the soil, they're in plant litter, they're in the plant rhizosphere. So all the things associated with plants, the plant phylosphere, marine environments, freshwater, engineered environments, stuff that we make, buildings and so forth. We find them there, aquatic, and also in animals. We have them in us. All of you have viroids in you, especially in your gut. They've been discovered. We don't know what they're doing. We have no idea, but they are there. And so I think the host range is far more than plants, but that remains to be discovered. Okay, so that's viroids. I think they are very cool, and the name is great. They're not viruses because they don't have capsids. That's like the defining feature of a virus. It has a capsid of some kind. So viroids are not. Diener called them viroids, which I think is a brilliant name. All right, next we're going to talk about satellites, or satellite viruses, not the satellites up in space. These are viruses that depend on a helper for replication. So they're viruses, they're in a capsid, they can't reproduce on their own. They have to co-infect the cell with a helper virus. And their genome can be single-stranded RNA, it can be DNA, it can be circular RNA. They do they can encode some proteins, but they don't encode proteins for replication. That has to be provided by the helper. And there are two kinds of satellites. There are satellite viruses that actually encode capsid proteins that will encapsulate the genome. And satellite RNAs don't encode their own capsid. They're packaged by helper virus protein. And these satellite RNAs might not encode any protein whatsoever. And so there's a picture of a helper virus capsid where you have a satellite RNA and the helper RNA co-packaged. These are different sizes. The class 1s are 220 to 1500. They have an open reading frame encoding non-structural proteins. Another class is, this one is linear RNA. Another class is linear RNA less than 700 bases, don't encode any protein. And class 3 are circular RNAs that don't encode protein as well. And again, these have to be replicated and packaged by a helper virus. And so when the satellite virus enters a cell, which is shown here, a eukaryotic cell, it circularizes, replicates by rolling circle by helper virus enzymes to make concatemers that are then cleaved, circularize, and go through this system over and over again. So they depend on the helper virus for replication, and then for packaging, many of them depend on the helper as well. So here's some examples of satellite viruses. There's some dependent on adenoviruses or herpes viruses. So this is adeno-associated virus, which will figure prominently on Monday when we talk about gene therapy using viruses or viral therapeutic viruses. There is a virus called AAV that will not replicate without either adenovirus or herpes virus co-infecting the same cell. And these are single-stranded DNA. They make particles, make a couple of capsid proteins. These have vertebrate hosts. And then here's another one, chronic B paralysis virus and the chronic B paralysis virus satellite. So that's the pair, right? So here adeno, AAV is the pair, CBPV satellite is the pair. These are single-stranded RNA viruses making particles. They have a small genome with one capsid protein. And obviously this one infects bees. Yeah, bees have viruses. Everybody has viruses. And bees can be wiped out by viruses, in fact. And then we have a plant satellite, tobacco necrosis virus, and TNV satellite. So tobacco necrosis virus is a serious pathogen of tobacco plants. So people worry about it because the world grows a lot of tobacco for people to use, right? To smoke and get lung cancer. So this is bad. And then the satellite modulates the disease. So this is the replication cycle of AAV. So this is a mammalian cell. It has AAV enters the cell, and adenovirus enters the cell. And adenovirus, here's the adenovirus genome in the nucleus, here's the AAV genome. You've seen both of these, right? This double T-ended single-stranded DNA, we've talked about how that replicates. There are a number of adenoviral proteins that are needed for AAV replication, including E1A and others. And we'll talk about that in more detail on Monday when we show how to design AAV vectors to do gene therapy. So already people have had their blindness cured by having AAV carrying the proper gene injected into their eyes, cured of their blindness. This is an amazing vector. It persists for many years. Now in plants, satellite cause distinct disease symptoms not seen with the helper alone. So helper plus satellite, you get a very different disease from the helper virus alone. And they often cause necrosis, which is cell death, or chlorosis, which is loss of chlorophyll in the plant. So you can see those leaves are turning yellow, and that's chlorosis, loss of chlorophyll. Now, one thing you have to understand is that satellites are not defective viruses derived from the helper. So they are defective, obviously, right? Because they cannot complete an infectious cycle on their own. But they're not derived from the helper. They have very little homology, if any, with the helper. They are just simply other viruses that have evolved to depend on a helper virus. I think what happened was one day a regular virus co-infected a cell with a virus that will become the helper, and that virus lost some genome sequence, but it could still replicate in the presence of the helper. And so that persisted. That defective genome persisted because the helper virus was there. Now we have a human virus that is a satellite and a viroid combined. It has properties of both. The helper virus is hepatitis B virus. And when you're co-infected, and you can only be infected with hepatitis delta virus, HDV, if you have HBV. You can't be infected with HDV alone because it needs HBV as a helper. It increases the severity of liver disease caused by HBV. So this is a seropositivity map of people who have antibodies against HDV and also have HBV infection. They measure that by antibodies against the hepatitis B virus surface antigen. And so the prevalence is shown here. The low numbers are the greens, intermediate yellows, and then the red are the highest. I think that's Mongolia, right? I think so. It's the highest percentage of people infected with both HBV and hepatitis delta virus. But you know, the US is 5 to 10%, South America countries, some countries are doing very well here. 18 million people currently estimated to be hepatitis delta infected. It's really hard to figure out exactly how many people are infected because we don't do a lot of screening. That's about 5% of the 350 million carriers of HBV. So this is the viral genome of HDV. It's a circular, the genome is negative stranded, and the antigenome is plus stranded. And it has a number of interesting features. First of all, it's highly base paired. In an infected liver, there are about 300,000 copies per cell, and it's 1.7 kb in length. It has a ribozyme, so it can self-cleave. That little red dot there is a ribozyme. And it encodes a protein called delta antigen, which is made from a single mRNA here. And this mRNA can give rise to two different size proteins, small and large delta protein. This delta protein is important for packaging. So on the right is the hepatitis B virus helper virus. And it's an envelope virus, of course, with glycoproteins in the membrane. And on the left is hepatitis delta virus. It's also an envelope protein. It has the envelope proteins of hepatitis B virus. So that's the helper function provided by Hep B. Envelope proteins to allow delta to attach to receptors and get in cells. And then inside you can see the delta genome, which is held there by a combination of large and small delta antigens. So the delta antigens are essential for packaging the RNA in the hepatitis B virus envelope. The replication of the delta agent is shown here. It looks a little complicated, but it really isn't. So we start with the genome, which is a minus strand, and then it is copied by host RNA polymerase to make it double-stranded. And this dot is the ribozyme, and so you can see at some point the ribozyme self-cleaves and you get mRNA encoding delta antigen. But sometimes it makes longer genomes which are circularized. So it makes concatemers instead of mRNA, and then those concatemers are self-cleaved by ribozyme and they make circular full-length plus strand RNA. And then the same process occurs to make the genome. It's simply copied again by RNA polymerase into concatemers, and the ribozyme resolves the concatemers so they can circularize and be genomes. Very recently, again, people looking at metatranscriptomic data. This is a new part of biology. All these sequences are available to you. You can go on your computer and find all these databases and just search them for stuff. And that's what people do, sit in front of the computer all day, and they find new sequences in these databases. So these were discovered in RNA transcripts of healthy birds and sick snakes. And a whole bunch of hepatitis delta-like viruses. So here, this is a phylogenetic tree of all the delta viruses. So at the top are the human delta viruses, which differentiate into different branches. And you can see the snake and the avian HDV-like viruses are quite off from the main cluster of sequences. So they've diverged quite a bit. What's interesting is there's no hepatitis B-like virus found in these birds and snakes. But there has to be some helper virus, right? Because it can't replicate without a helper. And people have subsequently found that, at least for hepatitis delta virus, if you put it in cells in culture, other viruses can serve as helper viruses besides Hep B. Hepatitis C virus, VSV, dengue virus, and West Nile virus can all provide a capsid for delta virus. So it's not just Hep B, although in people we don't see any other helper virus, but it may be that in snakes and birds and other animals, they use other viruses as well. Quite interesting. Now there's a set of viruses that are related to satellites called virophages. So phagein is Greek for to eat. So when bacteriophages were first discovered, right? They were shown to kill bacteria, so they were called phages because of this Greek word, virus eater. But the virophages eat viruses. So bacteriophages eat bacteria. The virophages eat viruses. I'll show you what I mean in a minute. They are circular double-stranded DNA virus with an icosahedral capsid, and they will replicate only in cells infected with a giant virus. They don't replicate within the giant virus. Some people say that all the time. The press says that. No, they don't replicate in a virus. They replicate in a virus-infected cell. And as I said before, they interfere with helper virus replication, which doesn't make any sense to me, but it works. These are out there in the environment. There are lots of virophages that have been discovered. You can see on this map, they're all over the world. You may be familiar with these ships that go around the world and take water samples at different depths and then sequence everything that's in it. And so you can search those and you find virophage sequences. And the hosts tend to be varied. So the first virophage discovered was called Sputnik, found in a cooling tower in Paris. It infects amoeba, acanthamoeba polyphaga, that are also infected with mimivirus, a giant virus. And then we have a virus from coastal waters of Texas that infects a marine phagotropic flagellate, and then lots of other viruses that are found in different parts of the world that infect different hosts. So I like this one here, lentil virus. It means lens in French. It was isolated from the contact lens fluid of a patient with keratitis in France. So they called it lentil virus. We would have called it lens virus, which is not as nice, right? Lentil is very nice. So we don't know what these are doing. We don't know if they're beneficial, but there is evidence that they move genes around from host to host and virus to virus. Here's what they do to interfere with their helper. So on the right is the structure of Sputnik, the first virophage discovered. Here are cells co-infected with Sputnik and mimivirus. So mimivirus is the helper virus. So I don't know if you remember, mimivirus is a giant virus with this fuzzy coat around it. And you can see that all of these EMs show defective mimiviruses, right? So this one has layers of defective protein around it. So does this one. These have actually Sputnik particles in the particle, and there's no genome. So this is disrupting replication of the helper. It's reducing the titer of the helper, but it does very well on its own, sufficiently to move on to a new cell. So I originally said another satellite is the virophage. And the people who work on virophages don't like them to be called satellites. They want to call them virophages or viruses. And here's the logic. All the other viruses we've talked about in this course depend on the transcriptional machinery of the host, or many of them, not all of them. Some of them encode their own transcriptional machinery of the host. But in this case, the viroid depends on the transcriptional machinery of another virus. That's the helper function that is provided. So they say, what's the difference where the transcriptional machinery comes from, the host or another virus? It's a virus, and I think that makes sense. We've answered the question so far. You can have an infectious agent that doesn't encode any protein, and now we're going to explore whether you could have an infectious agent without any nucleic acid genome, and this is Prions. Look at this. I found this sign on the internet. Somebody lives on Prion Road. Isn't that cool? Prions are infectious proteins with no nucleic acid. And they cause diseases like bovine spongiform encephalopathy or mad cow disease, Creutzfeldt-Jakob disease in people, scrapie, kuru, chronic wasting disease of deer and elk. And the one scientist who was instrumental in our understanding of what these are was Stanley Prusiner, who got the Nobel Prize in 1997 for discovering them. Now, I don't know if any of you are ballet fans, but George Balanchine was the director of the New York City Ballet years ago. And, you know, this is a man who used to be a ballet dancer and obviously could move his body really well. And suddenly, at one point, he had no longer been dancing. He started to lose his ability to walk, and he couldn't think properly. It turned out he died of a prion disease. And this is an article in the New York Times where they talk about how some of his brain slices were taken at Columbia Presbyterian Hospital. He died at 79, and they found evidence of prion disease in him. And this can happen just spontaneously with no known risk factor. So these prion diseases caused by these proteins are called transmissible spongiform encephalopathies, or TSEs. Encephalopathy is a disease of the brain, and what these are fatal neurodegenerative disorders of mammals. So only mammals as far as we know. Thousands of humans are diagnosed every year with TSEs globally, and about 1% of these arise by infection, meaning these people get some contaminated material either by eating or some medical procedure that infects them with these prions. And in a story that I will tell you in a bit, by 2002, 120 humans had contracted Creutzfeldt-Jakob disease, which is the name for a human TSE, by eating beef from animals that had mad cow disease. And this is because it doesn't matter how well done your hamburgers are, you can't destroy the infectivity of the prions. So TSEs occur in many mammals. They occur in cows. They occur in deer, elk, and moose. They have different names: Nyala, greater kuru, cats, both domestic and big cats. In sheep and goats, it's called scrapie, and it also occurs in mink. So a lot of animals get this disease. And in humans, we have a couple of different kinds of TSEs. Creutzfeldt-Jakob, fatal familial insomnia, Gerstmann-Straussler, kuru, and variant CJ, which is what you got if you ate contaminated beef from a cow with BSE. All of these are slightly different pathologies in the brain and slightly different symptoms. So that's why they've been named differently. I think the fatal familial insomnia is so scary, right? It's bad enough that you can't sleep, and then you die of it. And familial means it's inherited. So transmissible spongiform encephalopathies. The spongiform refers to the brain. As you take brain sections, they have holes in them, like you see here. Those holes are not supposed to be there. Your brain should be a nice, solid sheet. Okay, the holes are bad. That means cells are dying. And these individuals have severe psychomotor dysfunction. They have cognitive difficulties and all sorts of problems. And the different diseases can infect different parts of the brain, and they can give you different symptoms. And as I said, each disease has a characteristic symptomatology and pathology. The first TSE recognized was scrapie. We recognized the TSE in sheep because the diseased sheep would rub themselves on fences, and you can see here, that's why it's called scrapie, and they would lose their fur. I don't know what sheep have, fur or hair. What? Wool. Was it fur or hair, though? It's neither one. Okay. Wool, the wool. The wool is missing. That's right. You cut it off and you make a sweater out of it. They get motor disturbances. They tremble, tremblement du mouton, paralysis, weight loss, and they die within a month or two. And this has been seen in European sheep for over 250 years. It still exists in many countries. It's endemic, which means it's always there. And in the UK, 1% of sheep every year develop scrapie. They develop it spontaneously. They're not getting anything that's contaminated. They develop it spontaneously, which I'll show you in a moment how it happens. So the sheep farmers were smart. They found that the animals could transmit scrapie from one animal to another. They would give their neighbor farmers a sheep or two, or some sheep, exchanging sheep, right? And they noticed that the sick sheep infected the other. So it was an infectious agent, clearly. And in 1939, an experiment was done showing that if you take an infected sheep, you take the brain and grind it up, the infectivity will pass through filters that only let viruses go through, right? And so that's why for many years people thought this might be a virus, but it's really resistant to UV, ionizing radiation, formaldehyde. You can autoclave this thing, it will still have infectivity. Very hard to get rid of it. And so actually some people thought of the idea that there was no nucleic acid in it. It's got to be an atypical infectious agent. So here's an initial experiment that was done where we are looking at the inactivation of infectivity of different viruses by irradiation. So here's increasing dose, and this is the nucleic acid molecular weight. So the bigger the molecular weight of the viral genome, it's a bigger target size for irradiation, so you need less to break the genome. So herpes, the big viruses up here need much less radiation than smaller viruses like SV40. These are DNA viruses and these are RNA viruses. And when they did this with the scrapie agent and measured infectivity, it was way off the charts here. It's 10 to the 7th. You need almost 10 to the 7th dose to inactivate. So it didn't behave like any virus. I don't know why porcine circovirus is here. It doesn't make any sense. That's a single-stranded circular DNA virus. But obviously scrapie was different from all the other viruses, suggesting there's no nucleic acid genome to be inactivated.
Wool is a type of hair.
Wool is a type of hair. I know this because if you're allergic to hair, you get dogs who have fur instead. They don't shed. Early on, we realized that the animal and the human TSEs have similar histochemical abnormalities in the brain. They have all these defects that are listed here: plasma membrane formation, vacuolization, loss of neurons, giving you that spongiform appearance, accumulation of proteins in clumps, and amyloidosis, fibrils of misfolded prion protein. So people said, well, let's study the animal prion diseases to learn about what's going on in humans. And so what they would do is take organ homogenates from an animal who has died from a prion disease, inject them into a susceptible species, and then sometimes months or years of incubation would take before the disease becomes evident. And so people tried to find animals where the incubation time was lower, but you still need to wait around a year in the best model. And the way these prions work, you're injecting essentially prions from the sick animals. They begin to accumulate in organs in the target animals, and then eventually they go into the CNS where they cause that spongiform appearance. What's really interesting is that there's no inflammatory response, right? Remember, we've talked a lot about how viruses can induce inflammatory responses, which is a product of the innate immune response, but there's no inflammatory response, there's no antibody response, and there are no T cell, there are no cellular responses of any kind. So that tells us it must be a self-protein, and that's what it is actually. So the problem with TSEs is that you don't know you have them until you get symptoms, and then it's too late to do anything. They're untreatable, invariably fatal. There's no way to alleviate the symptoms. Nobody has any drugs that can help do anything with TSEs. So in 1967, a mathematician by the name of Griffiths said, I think these TSEs are actually proteins. And so Prusiner worked to try and purify the protein, and he purified infectious protein from the brain of sheep with scrapie. He purified the protein, and he injected it into animals and showed that it caused the disease, the TSE. And so that's why he called it a prion, because he purified the protein and showed that that caused the disease, and proteinaceous and infectious particle. And, you know, for years people said, oh, your preps are contaminated with some nucleic acid. That's what's actually causing the disease. He used to get up at meetings and talk, and people would actually get up and ridicule him. You don't know how to purify protein. Obviously, it's contaminated with nucleic acid. Well, in the end, he won the Nobel Prize, and they all had to shut up because he was right. The protein, prion protein, is encoded by a host gene in all of us, called the PRNP gene, and it's essential for TSE. If you don't have a PRNP gene, we don't know any humans who don't, but you can take it out of mice, you don't get TSE. So the current view of what a prion is that the prion is a conformational isoform of a normal host protein. So the normal host protein is called PRPC, prion protein C for cellular, and it's mostly found on the outer surface of neurons. It's GPI linked, it's not a transmembrane. And when you acquire, for example, an abnormal conformer, which we call PRPSC for scrapie, that causes the transformation of your normal prion proteins into the pathogenic form. So that's how it works. Once you have a little bit of PRPSC in you, then it converts all of your PRPC to PRPSC, and that's why you develop the disease. Remarkable. So here are some structural biochemical differences between PRPC and PRPSC. This is a schematic of the PRPC protein. It's mainly alpha helix with a couple of beta sheets here. And if you digest it with a protease called proteinase K, it is completely digested to peptides. The PRPSC protein has a lot of beta sheet character. And so when you cut it with proteinase K, it's mostly resistant to digestion. You get a little bit of cutting at the end terminus, but then you get this 27 to 30 kilodalton piece. And in the early days, that's what was used as a diagnostic. They would take a little bit of a biopsy from a patient to do a Western blot after proteinase K digestion. You could see whether the patient had this PRPSC or not. So the idea is PRPC is present in all of us, and it's converted to PRPSC. And how that happens, I'm going to show you. And really important part of this idea is that mice lacking the PRNP gene are resistant to infection. So you can knock out the PRNP gene in mice and inject them with prions, they will not develop disease. You need to have your own copy of the PRNP gene to develop a TSE. Now, how can you get a TSE? So you can get infected. You could eat something that's contaminated. You could get, in the old days, you could get a corneal transplant from a patient who had prions but didn't have any disease, right? And that cornea would then give you PRPSC, and you would end up with a TSE and die. Or other things purified from people contaminated with PRPSC. So that's infection. But you could also have mutations in PRNP that predispose you to misfolding. And whether you're infected or you have these mutations in the gene, the PRPSC begins to accumulate in the CNS, and that leads to your symptoms. So there are three types of spongiform encephalopathy. So the infectious or transmissible, that's where we acquire PRPSC, and it makes our PRPC misfold. And so we build up levels of PRPSC, and they cause the disease. Then we have familial spongiform encephalopathy. This is what you inherit from your parents. You inherit one or more mutations in the PRNP gene that at some point in your life suddenly make the protein misfold into PRPSC, and now that causes all your other PRPC to misfold as well. So you haven't gotten any infectious agent. It came from the mutations in your own gene. And then there's sporadic. You haven't eaten anything contaminated. You haven't gotten any contaminated transplants. You don't have any mutations in your PRNP gene. The protein just misfolds, and it causes all your other PRPC to misfold as well. So all three diseases can be transmitted to animals by inoculating the protein into them or having them eat it, because in the end, PRPSC is what causes the disease. So human TSEs can be infectious or transmissible and sporadic and genetic, as I'll show you. They include Kuru, spread of TSE by transplantation of infected corneas, hormones, transfusion from patients with CJD. So hormones now we make by recombinant methods, so this is no longer a risk factor. But you can see that if you get transfusion, well, now we actually have a PCR method to detect prions. But before we couldn't, and you would inadvertently give people contaminated blood. BSE was given to cattle by giving them infected sheep meat to eat. Talk about that in a moment. And then people got variant CJ from eating contaminated beef. So the first human TSE known was Kuru. It was a fatal TSE found in the 4 people of New Guinea, which is shown in red there, just above Australia. 30-year incubation period. Carlton Gajdusek, a virologist at NIH, went there to study it, and he found it was transmitted by ritual cannibalism of the brains of deceased relatives. So when someone died, the women and children would get to eat the brain. And if they die of a TSE, then the women and children get PRPSC and they develop the disease. So it was rampant throughout this collection of people. And he told them not to eat brains anymore, and now the disease is gone. Sporadic CJ happens in people with normal PRNP genes. It affects 1 to 2 per million people worldwide annually, mostly between 50 to 70 years of age. And this accounts for the majority of TSEs, 50 to 70%. So sporadic, no mutations, no infectious agent. They have normal PRNP genes. And you can transmit it to others if you develop a sporadic TSE and then you donate some blood before you know you're sick, you could spread it. And we think that this is how Kuru was established in New Guinea by that first person getting a sporadic TSE. They die and then their brains are eaten, and that starts the chain of transmission. Familial spongiform encephalopathy in people is an inherited disease, mutations in the PRNP gene. And again, if you develop one of these familial diseases, you can spread the agent to other people if you give blood or corneas or hormones and so forth. So again, all these ways that you can get a TSC, infectious, by mutation in the gene, by spontaneous misfolding, all have in common the PRPSC is converted to PRPC is converted to PRPSC. These are the deaths in the US by CJD from the 79 to 2020. So the red is the age-adjusted death rate. So you see it's pretty common between 1 and 1.5 per million people. And that's the total number of deaths over the years from CJ in the US. So if you ingest a prion, a misfolded prion, like in beef, how does that work? We don't really know, but we think there are prion proteins in the intestine, so you swallow it, goes into your intestine. There are PSCs on the mucosal epithelium. We think those are converted to PRPSC by whatever it is you ingest, and then somehow the PRPSC gets to the CNS where it causes more misfolding and eventually the disease. The genetic and the sporadic forms start in the brain, so there's no need to invoke this kind of alimentary tract spread. Now, BSE, bovine spongiform encephalopathy, was an outbreak of a TSE in cows in the UK, which was caused by forced cannibalism. Normally, you feed cows grass or corn, but if you want them to grow really fast, you grind up meat and you feed it to them, and then they grow really fast. No? You don't like it? Yeah, of course. But look what happened. And so here's a cow with BSE. What happened? So they used to feed cows, ground up other animals, including sheep who had died of scraping. And through many years, this was not a problem because the production method got rid of the infectivity somehow. But in the 1970s, the production changed. This is called meat and bone meal. That's what they feed the cows. And they allowed the scrapie proteins to pass to the cow and caused this outbreak of BSE. And they realized this and stopped this process, and the outbreak stopped. But many humans acquired a TSE by eating contaminated beef, and they got a new disease called variant CJD. And we estimate that 1 to 2 million of cattle were infected with prions during this outbreak. So these are this is the epi curve for the outbreak. So this is BSE in red, starting in the late 80s. So that's the number of cases of bovine on the left and human on the right. So the peak of the epidemic, 92, and then they changed the way the feed is processed, so the epidemic goes down. And then there's an overlapping outbreak in people of variant Creutzfeldt-Jakob, right, because there's a long incubation period, and eventually it goes down. But some of those people gave blood. They didn't know they were infected, and so these green bars are people who got variant CJ from blood transfusions from these people who got it from cows. The problem with cows is, at least cows that are meant for beef to eat, they're slaughtered in 2 to 3 years. They're big enough to kill. But the incubation period of the prion disease is 5 years. So you kill the cow before you even know it has the prion disease, and then the prions get in the food supply. Unfortunately, new cases of BSE still occur in cattle. They're probably sporadic. And they happen in the US, they happen in Canada and many other countries. But in the US and Canada, less than 2% of slaughtered cattle are tested. So the BSE could get back into the human food supply. And you can cook your hamburger for days and it will not inactivate the prion protein. There are some diagnostic tests. We could test more cows, but it's an expense, so nobody wants to do it. Unfortunately, these are the cases of BSE in North America. It's basically US and Canada from 1993 to 2018. There are 26 sporadic cases where they found a cow that was infected and they took it out of the food chain, fortunately. But I just wonder, people worry about this because it would just take one to get into the food chain. It would infect many people. Stay away from beef. So these are two really good articles. Top one is a guy who lived in the UK for a while and then moved to Saudi Arabia, I think, and then died of a BSE there, of a TSE, and it turned out that he had been infected while he was living in the UK, so a long incubation period. And then the bottom one is very scary, prion contamination in the emergency room. This is a story of a guy who is demented and shot himself, tried to kill himself, but he didn't succeed. He walked into an emergency room with brains leaking out, and it contaminated all the equipment, and they had to destroy hundreds of thousands of dollars of equipment because it can't be decontaminated. So now most ERs have protocols in place for determining whether someone has a prion infection. So they tested him, they saw he was prion positive, and they said, there's brains everywhere, we have to throw out all this equipment. Autoclaving, bleach, high pH, low pH, UV, they don't destroy infectivity. I did a podcast a number of years ago with Richard Knight, who's a prion expert, and if you want to learn more, it was very good. So we have had 3 epidemics of Creutzfeldt-Jakob disease in people. First, by injecting growth hormones purified from human pituitary glands. These people had TSEs, so there was a peak in the 90s, and we figured out what was going on. Transplantation of dura mater grafts, again from infected people, and then the one from cows, variant Creutzfeldt-Jakob, from prion-contaminated meat. 3 outbreaks of CJ, all caused by us, okay? All caused by us. So there's a cool assay for prions. It's kind of like PCR. Turns out that the prions are in the olfactory neuroepithelium, so you can take a swab up there, and then you put it in a tube. And so you put in the tube the swab material, and if it has some PRPSC, it's going to be oligomers. And so you put some monomers in, and you shake them, and the monomers are going to get bigger. They're going to convert, and the monomers are going to get bigger and bigger and bigger. You can sonicate it and do many cycles. And so basically a small amount of PRPSC is amplified, and you can easily detect it, say, on a Western blot. So this can be used actually with a swab and also with urine. All right, so the last thing I want to talk about is the prion species barrier. Usually you have to inoculate TSE, a prion, into the same species that it came from to cause disease. Going into the different species is inefficient. And so in other words, the sequence of the protein should be the same as you're putting in to the animal. And that's the barrier to interspecies transmission. So here's an experiment. You take hamster prion, PRPSC, you inject it into a mouse, there's no disease, right? It's not the right species. But if you put the hamster PRNP gene into mice as a transgene, then you infect them with hamster PRPSC, they get infected. So because now the hamsters have the same protein as the one you were inoculating. So this is useful. The problem, and I'll show you why, cow prions have a broad host range. Bovine prion, PRPSC, can infect mice and cause disease. And that's why probably their prions could infect us as well, even though we're the wrong species. So some prions can overcome the primary sequence barrier, and that's why we're worried about BSE, because it can infect us. Now there's another TSE that's been spreading lately, and that is chronic wasting disease of deer and elk and moose. So that's a deer with a TSE. You can see the characteristic signs. It's been detected in many US states and 4 Canadian provinces, in free-ranging cervids, which are deer, elk, and moose, and also captive commercial facilities. In some herds, 90% of deer and 60% of elk are positive. And in the wild, it can go up to 15%. All right, so these are a deer form of a prion disease. Here's where they've been found in the US. First, the epi curve, you can see it's been going up since we started looking for it in the 60s. And the more we look, the more we find. And these are the states where it has been found in free-ranging populations, captive facilities, and so forth. So the concern is as follows. So people have done experiments which show that if you put prions in the soil near plants, they will be taken up and displayed on the leaves, and that can be transmitted to animals and cause a prion disease. So you can imagine if these deer are excreting prions into the soil, the plants can pick them up and some other animal might get it. Maybe if it's a cow in a pasture, the deer have come in, and have contaminated it, it could transmit it to the cow. That's what we're worried about. But the good thing is if you take cervid PRP-SC, you put it in mice, no disease, it's the wrong species. If you take cervid PRP-SC and put it in mice transgenic for cervid PRNP, now you get disease. So the cervid protein doesn't cross the mouse species barrier. If you make mice transgenic for human PRNP, the cervid PRP-SC will not cause disease in those mice. So that's good, but things can change, proteins can change, and so who knows what's going on. So in the wild, we're concerned that the deer prions could spread to other animals. Shown here. They've been shown to infect cows when inoculated directly into the brain, but I don't know about oral feeding. So, none of these have been shown to be infected. However, a couple of weeks ago, a paper was published in Emerging Infectious Diseases showing detection of these CWD prions in wild pigs in Arkansas. So they captured a number of wild pigs. You can see the numbers here, about 100. And then they took out brain, retropharyngeal lymph nodes, and submandibular lymph nodes. And they did this amplification with a deer substrate. And they found a good fraction of the pig tissues positive. So somehow, and these are in areas where wild deer are infected with cervid prions. So somehow it's getting into these pigs. If you hunt, there's a website, cwd-info.org, which gives you instructions. Don't capture a deer that's sick. Wear gloves. Don't saw through the bone. Avoid the brain and spinal cord. Wash your hands. Don't eat the brain, spinal cord, eyes, spleen, tonsils, and lymph node. And certainly don't eat meat from an animal that tests positive, because we're worried about this for sure. So this is the overview now to end. We think that the original TSE arose in sheep spontaneously. Maybe that transmitted to deer to establish CWD, or maybe deer also developed a sporadic disease. But then from sheep, we went to cows, to humans, and to more humans. And these cows were also fed to other animals and who developed TSEs, including cats. It was put in cat food, and many cats developed TSEs. And then, of course, we have the spontaneous Creutzfeldt-Jakob, which could be transmitted to humans as well, as I have shown you. So that's TSEs. Okay, next time we're going to end this course by taking everything you know and turning it for the good. We're going to talk about therapeutic viruses.